All right. So good afternoon, everyone. I'm Michaela Pochner, and I'm the managing editor at Strip-tillage Farmer. Thanks for joining us today for the digital demonstration of Advancing Eco-Agriculture's Regenerative Soil Primer program. Today's presenter is David Miller, AEA's Director of Education. David has a passion to teach farmers how to regenerate their farms by capitalizing on how plants and soils interact. And as the leader of AEA's education effort, he directs ongoing education for new and existing AEA consultants. Before he gets started with the presentation, I just want to let you know that you'll see a control panel on your screen with a Q&A icon. You can submit questions here at any time during the presentation, and we'll take those at the end. So again, you can submit at any time, but we're saving all the questions for the end. And then we are recording today's session in the event you want to go back and watch it or share it with anyone. You'll receive a link with the recording via email within 48 hours of this presentation. So without further ado, I'll hand it over to David.
Thank you, Michaela, and welcome to all of you. Like always, it would be more delightful if you were all sitting around the conference room here and we could just, have a cup of coffee or drink and discuss these exciting things with each other. But since we don't have that luxury today, we will use the glamours of technology and talk about the Regenerative Soil Primer program. Advancing Eco-Agriculture was founded in 2006 and has since then become a national company working with growers of every size, from small gardeners to tens of thousands of acres, with almost any crop, especially any of the common crops that you want to name, we've probably worked with. So today, unlike normally when I do presentations where I talk about the science, we're going to specifically focus today on a key program, a program that's a keystone of the success that we've been able to help growers experience, and that's the Soil Primer program. And I was thinking about why we termed it the Soil Primer program in the first place. Why is it called that? And it's kind of like priming the pump. It's kind of like getting things started. It's become a very, very important part of the system that we put together for growers. So we're going to have a quick demonstration, if you will, of why growers have consistently come back year after year to get the soil primer and what they have been seeing with those things and how it can add value to your operation. So I'm going to go through this fairly quickly. There will be a little bit of sciencey stuff mixed in just to help you understand what the products are doing. And then I'm going to try and make sure we have plenty of time at the end for Q&A. So if you want to put the questions at the bottom, I believe it is, and there's a Q&A tab. If you want to do that, we will get to those at the end. So the Soil Primer program is made up of 3 different products: Rejuvenate, Spectrum, and Sea Shield. And on the right here, you can see what each of these products does. I'm not going to read that for you since you've probably done that already. What I really want you to remember is that Rejuvenate is food, shelter, and tools. So think of Rejuvenate as a food source for the bacteria and the fungi, whether that's the microbes that are existing in the soil and you want to support them, if you want to feed them, if you want to help them through a stress period. Rejuvenate can do that for the bacteria you already have in your soil. And then also very important for whenever you're inoculating biology, to provide a food source until your biology is established on the existing organic matter and or living plant roots. Rejuvenate also provides shelter, if you will, or places where the bacteria and the fungi are able to attach to and draw energy from, and then tools. And we'll get into that a bit later about how Rejuvenate provides tools for the bacteria and the fungi in their work of feeding the plants in that symbiotic relationship. So that's rejuvenate. Remember, food, shelter, tools. You want to know which of the products you should use or when you should use them. That's what rejuvenate is. Spectrum is microbes. It's the actual biological inoculant, and it's lots of microbes, lots of different species and subspecies of microbes. And this is something whenever you're looking at inoculating your soil, which you might think, well, there's lots of native biology out there, and there probably is. But when you think about all of the antimicrobial things that we've done in agriculture to date, there's actually quite a good possibility that a lot of species of microbes and even maybe keystone species are missing from the soil. So the spectrum is a broad array of and diversity of microbes and microbe subspecies, so that you're not just, and where I was going with the original thought of what to look for is to make sure that whenever you buy or inoculate with biology, whatever you're using, make sure that you have more than one or two or even 5 species of microbes, because if you just inoculate with 1 or 2, they're not going to be balancing each other out, and you can actually create an imbalance in the soil. And then the other thing to always consider with a microbial inoculant is the source of microbes. And with the spectrum, all of these microbes are grown as individual species and then put together in a dormant, dehydrated form. And so they're in a dormant state when you get them. They can be stored for a long period of time. I think 3 years is the expiration date. So as long as you keep them out of direct sunlight and away from anything that would be have negative effects on microbial spores, the spectrum is a very stable, very easy to use product, and very, very effective in establishing keystone species to help the soil regenerate and for other bacterial species to then be brought in by plant seed and biology, etc. Spectrum has, no, actually, I was looking at the wrong slide, or I thought I had moved already. What I want you to remember when you think of spectrum, and really the whole soil primer program, is carbon cycling, water efficiency, nutrient availability, and pathogen resistance. So when you think about microbes, bacteria, fungi, the whole microbial system, those are the 4 key things that we're really looking to accomplish, and that the combination of rejuvenate and spectrum have consistently performed. Spectrum has a subspecies or a sister product, if you will, or maybe it's a daughter product, called Spectrum DS. And so this is same as spectrum, but has a higher concentration of bacteria that are able to survive and to flourish, if you will, in drought and in high salinity areas. So Spectrum DS is the version of spectrum that we'll use if we're working with high sodium areas or in dry land situations. There's also a spectrum product called Spectrum PSB, which stands for phosphorus solubilizing bacteria. So again, same as the original spectrum, but has a higher concentration of bacteria to help solubilize phosphorus. And we could talk for a long time about how, well, maybe I'll just add a quick note there that when you look at a soil analysis, there's often, moderate to low levels of phosphorus. Some areas have very high levels of phosphorus. But when we look at a total nutrient assay and we look at all the phosphorus in the soil in the top 12, 18 inches, there's tremendous amounts of phosphorus out there. And often we're just missing specific microbial interactions, and either microbes being there or the microbes having what they need in order to break that tricalcium phosphate bond. And therefore, our plant sap analysis, or your plants are going to show that you need more phosphorus. So instead of just buying more MAP or more DAP, which in this, depending what region you're in, is an impossibility or is very expensive, think about using the phosphorus that's already in your soil. And in most regions in the US, there's sufficient levels of phosphorus to grow many, 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 many crops if we have the biology to do that. And then the Sea Shield component of the soil primer program. Sea Shield is a combination of fish, crab shell, and shrimp shell. So there's lots of ocean goodies, and for all of you who like seafood, well, microbes like seafood too, and they do really well with it. So with Sea Shield, I want you to be remembering fish oils. So think about oils as being very complex carbon molecules. So very stimulating for fungal growth and can be used as a fungal food and energy source. It provides amino nitrogen very effectively and efficiently. And then from all of the crab, the shrimp, and the fish, there's lots of minerals as well. So, what does the Regenerative Soil Primer do? It consistently boosts microbial populations. We see it very consistently accelerating crop residue and decomposition, and we'll talk a bit more about that, and you'll see and hear from growers who have been doing that. So if you're living in a region, in an area where crop residue cover is essential for overwintering and keeping your soil covered and keeping a carbon mat on your soil, you want to be careful about applying the regenerative soil primer too early, because you may lose more than you want to. It really works very well. Increasing cover crop success, and you can see on the roots at the right, this was a wheat crop, not a cover crop. But if you just look at the difference in the root structure, and this was the only difference here was Rejuvenate, Sea Shield, Spectrum. I do think these seeds might have been treated with the BioCode Gold. But if you look at the picture on the left, where we have the with and then the without, it is tremendous difference. You can see all the root hairs and how those are covered with a microbial sheath, which means that there's lots of bacteria and fungi that are secreting a compound called glomalin that will essentially glue these little dirt particles together into soil aggregates. And then because you have such a high concentration of this bacteria and fungi right on the root zone, that's why you see all of this soil sticking to the roots. And you have great aggregation going on there, which means you have good gas exchange, you have lots of water absorption. And if you look at the picture on the right, well, there's the soil structure is completely different, and then the root size and the interaction or the microbial interaction with the roots it's obvious that there's very little there, because those roots are just white, which means there's very little. They're having to really just pick up the simple ions out of the soil as root tips. So we could talk for a while about the rhizophagy cycle and how the rhizophagy cycle, which if you go onto our YouTube channel, you can learn more about that from James White from Rutgers University. Very, very fascinating how the plants farm the biology and how that enhances root structure and root branching. So these are all things that we see consistently with the Regenerative Soil Primer. So we're going to look at another scenario with roots, and then I'm going to talk for just a bit about the science behind Rejuvenate, Spectrum, and Sea Shield, and how they all work together to ensure that you have success in accomplishing these microbial establishments. Let's see if this works.
The root growth and the root mass was unbelievable in the 88 products. It's all about the roots. It's all about how many roots you can get off the plant. And what we saw was at least a 50% increase in root mass. To me, that translates into bushels per acre. The more massive root growth you get with a product or a system, translates into bushels. I think the biology is the key to what we're doing here. If we don't have the healthy biology, we can't move forward.
So true. So true. If we don't have healthy biology, we're not going to go anywhere. We're not going to move anywhere. It's not going to work for us. So what I want to look at now is if it were a sneak peek into being a microbe, and I wish I could do this better. In fact, if microbes lived a little bit longer, I'd probably want to go and be a microbe. But since microbes, a bacteria may only live like 20 minutes, some of those species, and so that doesn't sound quite as appealing. What we're looking at here is a glucose molecule, which is what happens. It's the end result of the photosynthetic process, and from here, protein synthesis happens. And the reason I'm bringing this in is because I believe it's important for us to understand what microbes need in order to work for us, in order to successfully perform these jobs that we're giving them. So I'm going to simplify this. If you're a chemist or a biologist, you'll definitely agree that it's simplified. But for me, and I hope for all of you growers, I found this helpful in just simplifying the process of what happens. So if you think of that glucose molecule as 6 links of carbon, 12 links of hydrogen, and 6 links of oxygen, a 24-link chain, and then by attaching, say, a nitrogen or a sulfur to that, it becomes an amino acid, or whatever other mineral forms different amino acids. And so you have 20 different amino acids, and each of these amino acids attached to each other is the beginning of a protein. So let's just say we have this specific amino acid. Let's say it's alanine, or whatever the specific amino acid is you want to name here. And now we're going to add another amino acid to it, and another amino acid to it, and another amino acid, and so on and so forth. And every time that an amino acid is added, we need an enzyme reaction in order for that to be catalyzed. Otherwise, the reaction would take a long time to catalyze. So you can see everywhere that I put this yellow dot is where we had an amino acid attached to an amino acid. And these are the enzymes that allow the amino acid to be attached to each other and cause these reactions to happen. When we have this, we have what we call a polypeptide. And now polypeptides are attached to polypeptides. And again, we need that enzymatic reaction. And every time that an enzyme is used, and an enzyme is not used up in this reaction, it's kind of like an open-end wrench, if you will. You can tighten many bolts and the wrench is not used up. But these enzymes are not able to function without enzyme cofactors, micronutrients essentially. And so every time that we put a polypeptide together and a polypeptide together and a polypeptide together, whenever that process is completed, it's now considered a complete protein, which is what insects are not able to digest. If they don't have the enzymes to break those peptide bonds apart and break that complete protein, into smaller pieces. Now, why is this important, and why am I bringing this up about photosynthesis and protein synthesis in a conversation about the soil primer? The reason I bring it up is to just give us a bit of a picture of, and to think about this in reverse, in that when we think about crop residue, for example, and the same applies for nutrients that are tied up in the soil matrix, where you have complex molecular structures and carbon complexes, the bacteria and the fungi don't have a mouth where they can go out and eat your crop residue. They don't have jackhammers or power tools where they can go and break the molecular bonds of the nutrients and the carbon complexes. So how do they do it? And it's also interesting to think about how big is a bacteria. I mean, it's very, very, very small. And so when you think about what changes that we see and expect to see from bacteria and fungi working, suddenly we actually should take a lot of care. It's like, wow, these little organisms really are doing a lot for their size. And the way they're doing this, the way they're breaking these proteins apart, if you look at like this corn, for example, the one on the bottom was not treated, the one on top was treated. And this picture was taken about 3 months after treatment. So you look at how much degradation has happened within this corn stalk. And what's happening here is, there's more bacterial and fungal species that have been introduced. So there's more team players, if you will. And also, it was complemented by enzyme cofactors. So when we think about this, is when the bacteria and the fungi want to break apart a molecule, they will secrete an enzyme. And you can think of that enzyme almost like a cutting torch or a welder. So enzymes can weld those chains together, or they can cut them apart, as in a cutting torch. So when bacteria, fungi, microbes are breaking down, decomposing, we call it, crop residue, or making nutrients available, they are continually secreting these. You could almost also think of them as acids, especially when you think about living plants and you think about, oh, we have a plant disease. Well, what is this plant disease? It is a bacteria or a fungi that's secreting an enzyme. And as long as that enzyme is able to perform its function, it will, in a sense, burn a hole into that leaf and allow the bacteria or the fungi to start feeding there. So the reason I bring this up is because Rejuvenate is, when I talked about tools, Rejuvenate is not just a food source. It's not just a shelter or a carbon source where the biology can get energy from very complex carbon substances and have the shelter and the porous spaces of humus. It also provides tools or enzyme cofactors, so that when these bacteria and fungi wake up and they start going to work, they have some tools to start working with and don't need to be scavenging all of those from their environment. And I believe that is one of the very key things that makes Rejuvenate so powerful and effective as it is because it's not just simple sugars. It's very complex carbohydrates. It has stable humic substances, and it has all these tools packaged into one easy to use package to use with your inoculation or for the bacteria and fungi you already have in the soil. So if you're doing a cultivation pass or you are needing to put out an herbicide and it's going to negatively affect the biology you have in the soil, you can come back in with some Rejuvenate and just stimulate that biology or feed that biology and help it recover from the stress. It's, you know, I've had this this thought about trying to inhabit an island. Like, we're inoculating this biology. Well, what if we were trying to start a community on an uninhabited island, and we just drop people off? And it's like, bye. Hope you can find water. Hope you have a way to, dig up some roots. Hope you can figure out how to build a house. It's kind of a hopeless situation. But if we drop the people off and we have a container of fast food, fries and burgers and soft drinks, it's like people don't need to do anything. They just start guzzling on all the fast food. And that would be analogous of just putting biology out or feeding your biology with simple sugars. So they just all get fat and they just sit around. And then after a while, it's like, hey, the food's all gone. What are we going to do? But everybody's feeling so sluggish and so out of it and maybe even sick that it's like, well, and so the community starts best when we can drop people off with basic tools, with a shelter, even if it's just a temporary shelter, and a bit of good stable food like beans and rice and things that will keep, that aren't too easy to just eat and that won't make people feel bad. So think about, maybe that analogy can help you think about when we inoculate, when we try to establish these communities in the soil, how are we doing that? And I'm not going to get into it today, but I do want to add here that adding mulch, doing cover crops, being careful about compaction, not smearing your soil and over-tilling, these are all things that play into this system and program. So you could be using the soil primer program and spray it on and say, well, I didn't get any results, if you are not following other practices. You might see a response, kind of like a flash in the pan, but then it goes away. It's like, well, where did that go? So as much as this program works, and I've been with AEA since 2008, 2010, and there is no program that has been as effective and efficient and constant as this primer program. If I could only have one tool, I would pick this one because it helps mineralize your soil. It helps give that plant the best opportunity to get established and to grow and to prosper. So we're going to look at a couple more examples of growers who used a version of or the whole soil primer program. And what you'll notice with these different examples is that there's a lot of diverse situations that it worked. And we can talk about why it worked. And if you have questions.
So here we are at another part of the orchard, looking at the fungal response that we're having in compost treated Bing trees. So compost was laid down on the soil surface in the tree line. And then right here, yesterday, seed shield was applied. And you can see that we're having the beginnings of fungal response. And what I really like here is you can actually see the moisture level. So you can see this band that goes down the center here. And to the right and to the left, the compost is darker because our weed boom that we use to apply the sea shield bands it on either side of the tree, and we don't hit the very center part of the trees because our boom would get whacked on the trees. And so we go in only a certain distance, and there's a dry spot that's left here in the center that you can see right here. But look at where our fungal response is. It's right where the sea shield and rejuvenate were applied. And again, this was applied here yesterday, and because it hasn't been very warm, and you can actually see where the compost was wetted down with that treatment, and you can see here the fungal response that we're getting in this compost treatment. Really exciting. In cherries, we of course want to see high fungal populations, and that has been achieved with our Rejuvenate and Sea Shield application here. And I just think this is a very, very nice example of us stimulating biology with that treatment of Sea Shield and Rejuvenate.
Wow. So what happens when you have that kind of fungi and that whole population that is going to be not oxidized into the atmosphere, but that's going to be run through the microbial system and build into stable humic substances that can last for a long time and feed the next generation of bacteria and fungi. So it's powerful what happens in crop residue digestion when we use the Regenerative Soil Primer program. Compaction down, and some of you may have seen this. This is an older document of where we applied this program that we're talking about, the Rejuvenate and Spectrum, and then you see the compaction layer the compaction is down in 2013. The untreated, the farmer standard in green was 340, and the area where this was treated was 213. And this is not just some random one-off sample. This was a combination of hundreds of different samples throughout the field that were averaged. So looking at 2014 then, the compaction dropped even a bit more, and the grower standard increased a little bit more. And then on the right of this slide, we have the difference between Rejuvenate and Spectrum and Sea Shield applied on triticale residue in the fall and not applied in the fall. And as I was saying earlier, if you need crop residue, if you need that soil armor, and especially if you have like triticale or beans or something like that, it will burn through that very quickly. So in some scenarios, you may be better off putting it on in spring or putting it on later in the fall. And our consultant team is very knowledgeable and well able to help you kind of sort through that and empower you with the knowledge so that you can make a good decision on that. Here
when the consultants actually brought out and said, "Hey, we want you to apply the soil primer," the initial shock of that is, wow, that soil primer is expensive. But when you get to the end of the season and you see what other inputs you saved versus that soil primer cost, it's cheaper. The entire fertility program on this cotton crop was less than $140. We started using the soil primer last fall, and we didn't tell our field labor that we had done anything different. They came to me and said, "Look at the difference in the way the texture of this soil is, and how much easier the crop comes out, and cleaner the crop comes out." AEA taught me really one of the biggest things that we can do is cut out unnecessary inputs. So especially in fertility, there's a lot of things that we were applying just because it was what Dad did and what Granddad did. And it doesn't have to be that way.
You've not met James Johnson, I'd encourage you to make opportunity. I was actually just on our weekly consultant call with him a couple hours ago, and I said, "James, what do you want me to say on the soil primer webinar?" And he's like, "Don't let the sticker shock keep you from using it." And that is, I think, a very good word, because the soil primer program is not cheap, but we are able to help you place it where it's effective and where you can do it what fits in your budget and what fits in your crop. But James is seeing so much in nutrient savings that it pays for itself again and again. Another grower from Pacific Northwest on the water infiltration.
I'm Dan McClure with Walla Walla Organics. The farm is about a 600-acre farm. We have organic and we have some conventional. Started working with the AA about 4 years ago. All of our crops are irrigated, and water penetration is a big deal. Our soils will tighten up and water just won't go in. But working with the AA products that we've been putting on, we found that soil's starting to open up, water's getting in better, and crops are responding nicely.
So I don't know where you are listening from, but there's many areas in the country that have either too much water or not enough water. And it seems there's few places that have just the right amount of water. And so both of those really need good soil aggregation. They need soil porosity. They need to be able to, if they get a rain after a dry period, where they're able to soak that in. And I would encourage all of you growers to think about how you can manage that and how you can set your fields up that when you do get those heavy rains, where you're prepared to soak that water in, keeping your soil covered, having that soil aggregation, and increasing your infiltration rates. Another example of this is a corn grower in Iowa, and I want to point out to you. So the story behind this is the before treatment in 2020 is a Haney analysis that was taken before the application was made, and then the 2021 column, the green column, was the same time of year after a corn crop, measuring what was the difference, what was the change in nutrient availability. So this is the Haney analysis that looks at the nutrients that are available. And just when you look at potassium, 36 parts per million versus 132 parts per million. Wow. And who said that you need to spend $50, $75, whatever your budget was to put potassium on? Look at the available phosphorus, huge number. Look at the water extractable organic carbon, more carbon available to your plants. This is from one application. You get all of these nutrients plus the increased carbon. Look at the nitrogen. Huge numbers, huge numbers. So get the biology working for you. If you don't have microbes on your farm, if you aren't being friendly to your bacteria and the fungi and the protozoa and the earthworms, if you don't have these critters on your operation, get them. They are worth their weight in gold. This is another example of nutrient availability on a vineyard in California. And again, this is not just a one-off sample. This is an average of 12 biweekly samples taken from 2 different blocks, the treatment versus a control block. And the treatment has 4 years of regenerative management. So this is not just one application of the Regenerative Soil Primer program. This is the Regenerative Soil Primer program spring and fall. Occasionally there was one in the middle of the season. There was a stress period. But then there was also foliar and fertigation applications of nutrients throughout the season based on plant sap analysis. So we were working to help build the whole soil microbiome and the symbiotic relationship between the plant and the biology. And you can see the difference. I mean, look at your calcium, 700 versus 400 of available calcium. Huge, huge differences. You look at the available N, 46 versus 18. Organic matter, 2.3 versus 1.3, 1 whole point difference. And then the soil health calculation is almost 3x of the control. This is a text that just came in 2 days ago. You look at, and it's a bit difficult to see, but hey, it's what the farmer sent. And this is the non-AEA is the cotton in the left module, and the AEA is 20. Or the module on the right is 20 acres of AEA cotton. So he's guesstimating probably 2 more bales in the right module, and that was from the soil primer being a part of the starter program. So he put a rejuvenate spectrum application right in furrow, right with the seed, and got the biology connected right as the seed was growing, and then it grew out with the plant in the soil. This next one is a soybean grower, and the field that he's harvesting here is normally a 40 to 45 bushel per acre soybean field. And he sent us this little video and said, this is unreal. And again, this was the Regenerative Soil Primer and then 2 foliar applications. So it's, again, not just the soil primer, but a work of the whole program. So just watch these screen numbers: 71, 68, 71, 77, 88, 90, 79, 76, 66, 83, 85. This is 97. This is on a field that normally they're happy with 50 bushel soybeans. If they get 60, they're doing really well, and he's easily averaging 70. So a very happy, very, very happy grower. He loves biology. This is a story you may have heard if you've been following AEA for any time, but if you're new to AEA and you have problems with weeds, especially thistles, listen in.
In fall of 2017, we fertigated the soil primer through our pivot onto a corn crop that was fully matured, and we had a cover crop underneath the canopy of the corn crop of Italian ryegrass, full coverage of the soil with that companion crop, and the soil primer with their growing roots and everything just did a really good job of starting off right away in a good way. And then the next spring, we planted soybeans into that corn. At planting, I saw the can thistle patches, and I thought, oh boy, we're going to have to deal with these can thistle again this year. I mean, these can thistle patches have been there for years and have been growing and becoming more numerous in the field. By the time I came around to cultivate the soybeans for the first time, the can thistle were gone or greatly decimated. I mean, there were few and far in between can thistle patches or plants in the patches, very few. And in that starter for those soybeans was the soil primer, which had been 25 grams of Spectrum, half a gallon of Rejuvenate, half a gallon of Sea Shield. And I'm thinking that application, or a combination of the fall soil primer application and a half in with the starter, is what did the can thistle in.
So why would biology do that to Canada thistles? I don't want to get too deep into the weeds on this, but when you understand what specific weeds are there for, then you understand that the nutrient availability has a huge impact or effect on which weeds are going to show up on your farm. And so by applying these 2 applications of soil primer, one in late fall in the standing corn and cover crop, and then a lighter version of it in the planter, what we believe has happened here on Ted's farm is there was enough energy from the soil biology that there was enough calcium, which is the most common reason why you have Canada thistles, is because you don't have enough available calcium. And so by increasing the level of microbial activity in the soil, these Canada thistles didn't really have a reason to be there. There was enough available calcium, and their purpose was served, so they were gone. Oops, going the wrong way. Sorry about that. All right, so regenerative soil primer. Just a couple wrap-up notes here. The soil balancing without biology Meaning, why are you putting out more phosphorus and more potassium and more calcium and more magnesium into a soil that is already has plenty of it out there? And the reason that your plants aren't taking it up has nothing to do with the amount that's out there, but has all to do with not having the symbiotic relationship with the biology, or certain species or certain types of biology being completely missing. I mean, it's kind of like if you have a football team and a couple of players are missing, the call goes out, but there's nobody there to receive the message. If the plant says, we need more phosphorus, and we've learned that plants are actually able to do that. Plants will send out specific signals to stimulate or to call phosphorus solubilizing bacteria and fungi to get to work. We need more phosphorus. And if the biology is there, it will respond. And so if the biology is not there, well, then your plant isn't able to take it up. So, and I had an experience in this where we did a soil sample. We put out a ton of rock phosphate and the soil samples didn't move because we were just putting out more ground up rock in a relatively dead soil, and the biology wasn't converting it into an available form. A foliar without a rhizosphere symbionts is just simply a dead end. If in season, and now this is a bit of out of context for late season here, but if you apply a foliar application, you're never applying a foliar to provide all of the nutrients that your plant needs. You're only providing a trigger, or if you will, an IV for that plant to be able to produce more sugars, feed more biology, and then that biology will return more nutrients, and that's why you have less loss of yield and you harvest more crop. But if you don't have that biology at the end of the roots to respond to that foliar application and the increased sugar that the plant puts down, then it's just a dead end. So again, it's very important that you have the foundation of biology. So establishing a microbial plant symbiotic relationship is the key to success. It is critical. Unless you are satisfied with a simple ion, water-soluble, glorified hydroponic system, where your plants are only taking up simple ions in a water-soluble form, which is a big problem when you get into drought. Because as soon as your moisture level drops, all of that nutrient in solution goes away. Your potassium, your phosphorus that's in the solution dries up. And if your plant doesn't have the symbiotic relationship with the biology, it's done right there. If it has a good functioning microbial system, then it's able to actually make nutrients available through the microbial system into a much deeper level of drought. BioCode Gold is not typically considered a part of the soil primer program, and yet BioCode Gold is the most effective thing that you can do if you're planting seeds to establish mycorrhiza and bacteria. So BioCode Gold is often used in conjunction with, if there's a cover crop, the Rejuvenate Sea Shield Spectrum are put in furrow and or sprayed on top of the soil and then incorporated or irrigated in. And the seeds are treated with BioCode Gold. So just putting it out here, not because it's typically a part of the program, but because it is one of the easy ways, very cost-effective ways that you can establish that biological symbiotic relationship. And they told me to put this in. We are running an offer right now through November 10th. If you would like to get started, just go ahead and call the office. I have this slide right here. Give our office a call, and we will get you set up with the Soil Primer program and get your crops and your soil set up for success. That's what you want. So with that, Michaela, we'll open it for questions. And like always, I talk longer than I expected to. So.
Well, it was all great information. And I think those testimonials especially are really great to see in these types of digital demonstrations. So appreciate that. We did have some questions come in, and one I think you briefly touched on. It's two parts. It says, Dave asks, can you apply the soil primer through a center pivot sprinkler? And what is the best way to do that?
Yes, the answer is yes. And the best way to do that is have an injection system at the center of the pivot. There's a number of growers that we work with that have come up with very automated systems. I think there's a company, I might need to double check on this. I think it's called Surefire or Sure something in Kansas that have a very good injection system. It's been working with our products extremely well. So I would definitely give them a thumbs up, and I'll be happy to, if you want to reach out to the hello@advancingecoag.com and just mention or ask your question directed to me, and I'm happy to get exact details on that. But yes, as far as the application, it's very effective through the pivot. The only thing that I would say about applying it through the pivot is don't apply it on a hot, windy day when it's going to be dry and you're not having. So if you're applying it through the pivot in a dry scenario, then it's important to have enough water to take that bacteria down into the soil profile so it's not becoming dehydrated and you're not getting establishment.
Okay. And then Jeremy says, the common knowledge here is that we all need to do is spray some 28% N on our corn stalks if we want them to break down. How does that process compare with your soil primer?
Great question, Jeremy. Great question. And thank you for bringing that up. Actually, I meant to touch on that just a bit. What happens in that scenario is you have a very wide carbon to nitrogen ratio in your crop. And so when you apply nitrogen and you narrow that window, you create a situation where the carbon is oxidized and a lot of the carbon is lost into the atmosphere. So when you use the soil primer program, you're giving that biology. And now if you have you're giving that biology, you're allowing the biology to convert it into a stable humic substance that stays in the soil and isn't oxidized off as CO2. So just putting out the nitrogen, you're putting out nitrogen that you don't need at that time. And in this day and age, nitrogen's not cheap. So putting out nitrogen now is actually, you're just getting nitrogen, you're losing carbon. Whereas if you put on biology and a food source for the biology, as you saw on a number of the customers' experiences, you're getting a lot of nutrients like calcium and magnesium and micronutrients are becoming more available. So you get a lot more than just nitrogen, and the breaking down of crop residue is not a problem.
Okay. We have time for a couple more questions here. If anyone wants to submit, while people are thinking, I'll ask from your perspective, David, if somebody is interested in this program, what does it look like from step one, the grower telling you they're interested, to working with you, and how involved are you guys through the grower's time of using the product?
Great question. So our consultation and the way we work with growers is as customizable and diverse as probably the microbes in the soil, because we do really cater for growers for where they are. But let me just give you a common example. If you call the 800 number, you will talk to Stephanie or Katie or John, and they will ask you a couple questions. If you know exactly what you need and they're able to answer your questions, they can take your order. and we'll get it shipped to your door. We'll help you with any kind of application questions you have. If you're looking to do more of a whole farm system, or you have a specific scenario, they will consider your location, the crops that you're growing, and they'll connect you with a consultant who will spend 30 minutes to an hour evaluating your situation and giving you consultation, which is included with the product. And then if you decide to work with a grower, we cater to the grower's needs. I have growers that I talk to every week. We look at 20 sap analysis excuse me, every week, and we build foliar programs. We consider the soil. We look at soil analysis. We put on soil amendments, the recommendations, and I have growers that I talk with in spring and fall, and it's like, how's your crop doing? We spend a little bit of time, so we can really customize based on what growers' needs are.
That's great to hear. I also want to put in a little plug here. David and AEA did a webinar with our sister publication, No-Till Farmer, not long ago, and we recently posted an article on the No-Till Farmer site that talks about what David mentioned about those soil health practices that support the regenerative primer program, regenerative soil primer program, excuse me. And so if you're interested in that aspect of it, you can go back and watch that too. I think it's, like David said, it's a system that all works together. So that's available for anyone who's interested as well.
That's great. Thank you for mentioning that. And as amazing as the Regenerative Soil Primer program is, the combination of these three products, and as you saw, people were applying it in furrow at 2 quarts per acre of Rejuvenate, 2 quarts of Sea Shield, and 25 grams of Spectrum, which is half of our recommended broadcast application when you're planting it with like wheat or beans where you have narrow rows. And what's happening is you are putting this bacteria right with the seed, and then there's a symbiotic relationship that happens as soon as that root emerges. And then every root from there on out is just feeding the biology, and the biology is growing with the root and spreading across your soil very quickly. So depending on your scenario, you're able to get very significant effects with actually pretty low rates. In other scenarios, depending if you have a lot of crop residue, you're spreading it out across the whole acre, you don't have living roots, you might want to go to 6 quarts of Rejuvenate or 2 gallons of Rejuvenate. If you're in orchard and you have a problem with a certain insect or a specific disease like fire blight, for example, then you might want to go 2.5 or 3 gallons of Rejuvenate and double rate of Spectrum, or maybe you even want to switch to the biodigester, which is for very lignified material, and get it broken down before next season so that you don't have you're not hosting insects and diseases over winter. So the program is very diverse. But what my thought is that as effective, as powerful, as exciting as all of these results are, it's not a silver bullet. It does not mean that because you put on the Regenerative Soil Primer that bam, voila, right now you suddenly have magically created soil structure in that first 2 months. You still need to care for the biology. You still need to be thinking about the whole system. Now, I've seen that happen. I've seen that happen very often where it's like, wow. In fact, we had a consultant in Michigan that used to have growers split a field, and then he'd walk the field the following spring, and he had a very high accuracy rate of being able to tell which side was sprayed with the soil primer because of how soft the soil was. So it works. That's why you need to call the 800 number and get started.
Yes, and if you have any questions, call that number or send AEA an email there. But in the interest of time, I know everyone's super busy at this time of year. We're right at our end mark, so I just want to say thank you to David for sharing your expertise today, and thank you to everyone who joined us during this, like I said, very busy time of year. As a reminder, today's presentation and audio will be made available on the Strip-Till Farmer website. If you'd like to review what was talked about today, or show it to anyone that you know who might be interested. You will receive an email within 48 hours with the recorded presentation, so keep your eyes out for that. On behalf of David, Advancing Eco Agriculture, and Strip-Till Farmer, thank you again for joining us for today's digital demo. You can reach out at any time to let us know what you thought of today's live event and if there's any topics you'd like to see us cover in future webinars. So thanks again, everyone, and have a great rest of your day.